I had this massive breakthrough the other day. It completely shifted how I view my own existence, honestly.
Oh, yeah. What kind of breakthrough?
Well, we look in the mirror, right, and we see this solid, static thing, a single object, a body. We brush our teeth, we comb our hair, and we treat ourselves like one unified organism.
Right. You just see the macro level.
Exactly. But if you actually zoom in past the skin and past the tissue, you realize the human body isn't a solid block at all. It's a hyperactive, microscopic metropolis.
That is a great way to look at it.
Yeah, because you are walking around with roughly 37 trillion individual living cells. 37 trillion tiny houses just all packed together. And just like any massive, sprawling city on Earth, it absolutely cannot survive without a non-stop, hyper-efficient logistics network. You know you need grocery deliveries coming in, and you need trash pickups going out 24 hours a day.
That is the exact right mental framework to start with. Welcome to the deep dive. For you, the listener, our mission today is to completely map out this internal highway system. We were talking about body fluids and circulation.
It's the ultimate logistics map.
It really is. And I've spent over 15 years mentoring students and teaching this specific biological machinery. The biggest hurdle people face, especially in exams like Neet, is treating it like a static list of Latin terms.
Oh, yeah, just memorizing flashcards.
Exactly.
Yeah.
And it isn't that at all. It's fluid dynamics, it's electrical engineering, and it's brilliant plumbing. Every single one of those 37 trillion cells needs constant grocery deliveries, oxygen, glucose, amino acids. And just as importantly, the moment those cells consume those groceries, they produce toxic waste and carbon dioxide.
Right, and if the trash piles up,
the local environment turns acidic, the cells choke, and the city collapses. It's that simple.
So the overarching question for me, at least, the why does this even exist, comes down to scale and distance. Like if we were single-celled organisms, we wouldn't need a heart.
Exactly. Simple organisms like sponges or coelenterids, think jellyfish, they don't have blood. They don't have a heart.
They just use the ocean, right?
They literally just use the ocean. They pull water from their surroundings, circulate that water right through their hollow body cavities, and let their cells directly exchange oxygen and waste with the environment. It's diffusion at its most basic.
It's like living in a cabin right on the edge of a pristine river. You just dip your bucket in when you're thirsty and toss your waste downstream. You don't need indoor plumbing.
That's a perfect analogy.
But humans aren't cabins on a river. We are Manhattan. A bone cell buried deep inside your femur or a neuron locked inside your skull is, microscopically speaking, miles away from the outside world. It can't just absorb oxygen from the air.
Right, because diffusion is incredibly slow over long distances. If you relied on simple diffusion to get oxygen from your lungs to your toes, it would take years.
Years? That's insane.
Literally years. So as life evolved into complex, multi-layered organisms, we had to internalize the ocean. We had to evolve highly specialized internal fluids to transport materials rapidly under pressure. For humans and most higher organisms, that primary fluid is blood. And working right alongside it is a shadow network, a crucial fluid called lymph, or tissue fluid.
Okay, I want to build a really clear mental map of this. Because when you first look at the makeup of blood, it's classified in a way that feels completely counterintuitive. The biological classification for blood is a special connective tissue.
It throws everyone off the first time they hear it.
Yeah, because when I hear connective tissue, I think of tendons, ligaments, cartilage, stuff that physically binds bones and muscles together. But blood is a liquid.
Right, but in histology, which is the study of tissues, a connective tissue is defined by its structure, not its solidity. A connective tissue is essentially any group of living cells suspended in a non-living extracellular matrix.
Okay, so the matrix is the key.
Yes. In bone, that matrix is calcified and hard. In cartilage, it's rubbery. In blood, that matrix just happens to be a liquid.
So if we picture our mental map for you listening, blood is divided into two primary columns. The liquid matrix, which we call plasma, and the formed elements, which are the actual living cells and fragments floating around in that liquid.
Let's start with the river itself, plasma. Plasma is the foundation. It makes up roughly 55% of your total blood volume. Imagine if you drew a vial of blood and spun it really fast in a centrifuge. The heavy cells would get forced to the bottom, leaving this translucent, straw-colored, slightly viscous fluid sitting at the top.
That's the plasma.
That's plasma. It's sticky, it's slightly heavier than water, and it is a complex chemical soup.
But mostly it is water, right? I mean, the breakdown is something like 90 to 92% pure water, which makes perfect sense for a river. You need a solvent.
Precisely. But it's what's dissolved in that remaining 8 to 10% that makes it magic. Let's focus on the proteins, which make up about 6 to 8% of the plasma. In my years of teaching, this is a classic spot where people get tripped up. Examiners love testing this.
The big three proteins.
The big three. You need to know fibrinogens, globulins, and albumins. They each have distinct, non-overlapping jobs, and it is vital to keep them straight.
Okay, let's break them down. Let's start with fibrinogens.
Fibrinogens are the emergency repair materials. They are large proteins absolutely essential for the coagulation or clotting of blood. We will dive deep into the mechanics of clotting later, but for now, think of fibrinogens as undeployed nets floating in the river.
Undeployed nets.
Right. If a blood vessel gets cut, these proteins are activated to physically polymerize into a fibrous mesh to plug the hole.
Okay, undeployed nets for clotting. Got it. What about the second one, globulins?
Globulins are your defense department. Specifically the immunoglobulins, you probably know them better by their common name, which is antibodies.
Oh, okay.
They are Y-shaped proteins that patrol the plasma, actively searching for foreign invaders like viruses or bacteria to neutralize. So globulins equal defense mechanisms.
Fibrinogens for clotting, globulins for defense, and the third is albumins. What do they do?
Albumins are the smallest but most abundant of the plasma proteins, and they do something fascinating. They maintain osmotic balance.
Osmotic balance. Let's unpack that. Osmosis is water moving to where there's a higher concentration of stuff, right?
Exactly, because your blood is under pressure from the heart. Pumping the water inside your blood vessels naturally wants to get squeezed out through the microscopic pores in the vessel walls and into your tissues.
Because of the hydrostatic pressure.
Yes. If all the water left your blood would turn to sludge and your tissues would swell up like balloons. Albumins act like molecular sponges. Because they are large proteins trapped inside the blood vessels, they create an osmotic pull called oncotic pressure. This pulls water back into the bloodstream, counteracting the blood pressure, pushing it out.
That is brilliant. So if you have liver damage, since the liver makes these proteins, and your albumin levels drop, you start retaining fluid in your tissues.
That is exactly the clinical manifestation. Edema.
The swelling is literally the water escaping your blood because the albumins aren't there to hold it in.
You've got it. So to recap the big three, fibrinogens, clot, globulins, fight albumins, hold the water in. Aside from the proteins, the plasma is also carrying your basic minerals, sodium, calcium, magnesium, bicarbonate, chloride. Plus, it's the actual transit route for the groceries we mentioned.
Glucose, amino acids, lipids.
All dissolved or suspended here in transit to the cells.
Now here's where we hit a massive conceptual trap. I know people use the words blood plasma and serum interchangeably in casual conversation. You hear truth serum or blood serum, but biologically, serum is a very specific, isolated thing.
It is a critical distinction, especially in diagnostics.
Yeah.
Think about the plasma we just described. It's full of fibrinogen ready to clot at a moment's notice. But in a healthy, uninjured vessel, that fibrinogen is inactive. If you take plasma and you chemically remove all of those clotting factors, strip the fibrinogen right out of it, the fluid you are left with is called serum.
Here's what you must remember if you are listening. This is a golden formula to lock into your brain. Serum equals plasma minus clotting factors.
That is the formula. It's a classic Neet trap because it sounds almost too simple. If a lab wants to test your blood for antibodies, say, to see if you have immunity to a specific virus, they often don't want the blood to randomly clot in the testing machine. So they let it clot first, remove the solid clot, and test the remaining clear fluid. They test the serum.
Okay, so that's the liquid river, the 55%. Let's shift to the other 45% of our mental map, the formed elements. This is the cargo, the security teams, and the repair crews actually suspended in the plasma: erythrocytes, leukocytes, and platelets. Let's start with the undisputed kings of the bloodstream. Erythrocytes, or red blood cells, the RBCs.
They are the absolute most abundant cells in the human body. To grasp the scale, a healthy adult has on average 5 to 5.5 million red blood cells in a single cubic millimeter of blood.
A cubic millimeter is roughly the size of a grain of coarse sand. 5 million cells in a grain of sand. That is just staggering.
And every single one of them is manufactured in the red bone marrow deep inside your larger bones. But what truly makes the red blood cell an evolutionary masterpiece is what happens as it matures.
This was a genuine whoa moment for me when I first understood it. We are taught that every cell has a nucleus, right? A brain, a control center containing its DNA. But in humans and most other mammals, as the red blood cell matures in the bone marrow and prepares to enter the bloodstream, it actively destroys its own nucleus. It ejects it. It essentially commits a form of biological lobotomy on itself.
And it doesn't stop at the nucleus. It ejects its mitochondria, its ribosomes, almost all of its internal organelles. It empties itself out completely.
Why on earth would a living cell do that? Without a nucleus, it can't divide. Without mitochondria, it can't even use the very oxygen it carries for energy.
Because of supreme specialization. The entire singular purpose of an RBC is to transport respiratory gases. Every organelle it keeps takes up valuable cargo space. By jettisoning its internal machinery, it maximizes its internal volume. It then packs that empty space to the brim with a complex iron-containing protein called hemoglobin.
Hemoglobin is the actual molecule that binds to oxygen, and the iron inside it is what gives blood its red color.
Precisely. A healthy adult has about 12 to 16 grams of hemoglobin in every 100 milliliters of blood. Think of the RBC as a microscopic bus, and the hemoglobin molecules are the seats. By emptying out the engine and the steering wheel, you can fit more seats on the bus. Furthermore, ejecting the nucleus changes the physical shape of the cell. It collapses inward, becoming biconcave.
Like a donut where the baker didn't punch the hole all the way through. It's thicker on the edges and thin in the middle.
That biconcave shape does two incredible things. First, it drastically increases the surface area to volume ratio, which means oxygen can diffuse in and out of the cell much faster. Second, it makes the cell incredibly flexible. The smallest capillaries in your body are actually narrower than the diameter of a red blood cell.
Wait, really? The pipes are smaller than the trucks?
Yes, the capillaries can be 5 micrometers wide, and an RBC is about 7 micrometers across. So the RBC has to physically fold itself, squeeze and contort to slide through these tiny vessels in single file. That biconcave shape allows it to bend like a fluid-filled balloon without popping.
But there is a massive evolutionary trade-off here, right? Because they gave up their nucleus and their DNA, they have no blueprints for repair. If they get damaged squeezing through a tight capillary, they can't fix themselves.
It's a tragic existence.
Yeah.
They are born, they work relentlessly, and they slowly degrade. Because they lack repair mechanisms, their lifespan is strictly limited. An average red blood cell lives for about 120 days. After 4 months of constant circulation, the cell membrane becomes brittle and fragile.
And what happens to a brittle cell going through a high-pressure system? It eventually breaks. Where do they go to die?
The body anticipates this. We have a dedicated filtration organ, which is the spleen. The spleen contains incredibly tortuous, tight microscopic mazes. Young, flexible RBCs can bend and squeeze through the spleen's maze, but old 120-day-old brittle RBCs get stuck. Once they get stuck, special immune cells called macrophages devour them, breaking them down to recycle the precious iron and amino acids.
So the spleen is literally the graveyard of the red blood cells. That's a morbid but incredibly efficient recycling program.
Very efficient.
Okay, so we have our millions of oxygen-carrying cargo ships. Now let's look at the defense force. Leukocytes, or white blood cells. The WBCs.
WBCs are a totally different breed. First, they are colorless because they contain no hemoglobin. Second, they are true cells. They retain their nucleus and all their organelles. And third, they are vastly outnumbered by the red blood cells. While you have 5 million RBCs in that grain of sand volume, you only have an average of 6,000 to 8,000 WBCs.
That's a huge drop-off.
They are a highly specialized, elite, standing army, and they are generally quite short-lived, sometimes only surviving a few days or hours during an active infection.
When you look at them under a microscope, biologists historically divided them into two main categories based on their appearance: granulocytes and agranulocytes. Basically, do their insides look grainy or are they smooth?
The grains are actually tiny vesicles packed with chemical weapons and enzymes. The granulocytes include neutrophils, eosinophils, and basophils. The agranulocytes, which have clear cytoplasm, are the lymphocytes and monocytes. Now, learning all these names and functions can feel overwhelming. You need a structural hierarchy based on how abundant they are.
I love memory frameworks. What's the standing army look like in terms of numbers?
Neutrophils are by far the most abundant. They make up 60 to 65% of your total white blood cells. On the absolute opposite end of the spectrum, basophils are the rarest, making up just 0.5 to 1%.
There's a classic mnemonic for this, to remember the order of abundance from most to least. Never let monkeys eat bananas. Neutrophils, lymphocytes, monocytes, eosinophils, basophils, it lines up perfectly with the percentages.
It's an old, reliable trick, and it works. Now let's rigorously match the cell to its exact function, because this is where examiners test the nuances of the immune system. Let's look at the Pac-Man first.
That would be the neutrophils, which are 60 to 65%, and the monocytes, which make up 6 to 8%.
Yes. Despite being in different structural categories, neutrophils and monocytes share a core tactical function. They are both phagocytic cells. Phagocytosis literally means cell eating. When bacteria breach your skin, the neutrophils are the first responders. They physically crawl toward the chemical scent of the bacteria, envelop them, pull them inside their own bodies, and dissolve them with acid and enzymes.
It's microscopic hand-to-hand combat. And a gross but fascinating fact. When you get a cut and it forms white pus, that pus is largely just millions of dead neutrophils that gave their lives eating bacteria to keep you safe.
Exactly. They are the infantry. Now let's look at the rarest ones, the basophils at 0.5 to 1%.
If neutrophils are the infantry, basophils are the chemical warfare specialists.
Fight on. Basophils are heavily involved in inflammatory reactions. They don't usually eat bacteria. Instead, when they detect damage or invaders, they degranulate. They dump their chemical payloads into the blood. You need to know the three main chemicals they secrete: histamine, serotonin, and heparin.
Histamine is famous. If you have allergies, you take an antihistamine. What does histamine actually do physically?
Histamine is a vasodilator. It makes your blood vessels widen and become leaky. If you twist your ankle or get stung by a bee, the area gets red hot and swollen. That's basophils releasing histamine, opening the floodgates, so more fluid and more white blood cells can rush into the tissue to heal the injury. The swelling is a feature, not a bug.
That makes so much sense. Okay, next up are the eosinophils sitting at 2 to 3%.
Eosinophils are the specialists. Their primary job is resisting infections, but their clinical hallmark is their association with allergic reactions and parasitic infections. If a patient comes into a clinic and a blood draw shows a massive spike in eosinophils, the doctor immediately suspects they are either having a severe allergic response or they have a parasitic worm like a tapeworm or hookworm. Eosinophils are specifically designed to attack parasites that are too large for a single neutrophil to eat.
And finally, the strategic commanders. The lymphocytes making up 20 to 25% of the WBCs.
Lymphocytes are the masterminds of your specific adaptive immune response. We categorize them into two major types, B forms and T forms. T cells often act as the generals coordinating the attack and killing infected host cells, while B cells are the weapons factories. B cells are the ones that actually manufacture those Y-shaped globulin antibodies we talked about earlier in the plasma section. They are the reason you build long-term immunity to a virus after you've been exposed to it once.
It's an incredibly coordinated system that covers the RBC cargo ships and the WBC defense force. The final formed elements are the platelets, also called thrombocytes, the repair crew.
Platelets are a biological oddity because, just like RBCs, they aren't true complete cells. But they don't start as cells and lose their nucleus. They are born as fragments. Deep in the bone marrow, there are these massive, giant cells called megakaryocytes.
Megakaryocyte, giant nucleus cell.
Exactly. The megakaryocyte grows huge, and then it essentially shatters. It pinches off thousands of tiny pieces of its own cytoplasm and membrane and dumps those fragments into the bloodstream. Those fragments are platelets.
And they are highly concentrated. A normal blood count contains between 150,000 and 350,000 platelets per cubic millimeter.
Their singular mission is coagulation. They are packed with a variety of substances that, when released, initiate the clotting cascade. If a patient's platelet count drops too low a condition called thrombocytopenia, the primary risk isn't infection, it's hemorrhage. They can suffer excessive dangerous blood loss from microscopic trivial injuries that happen to our vessels every day without us noticing.
Which perfectly bridges us into the actual mechanics of plugging a leak, coagulation. Imagine if you get a tiny paper cut. In a high-pressure liquid system, like our circulatory system, why doesn't all the fluid just leak out of that hole forever? How does the body actually patch a pipe while the water is still running?
It is a phenomenal biochemical mechanism. When you get a cut, you bleed for a minute, and then the blood gets viscous, and eventually a dark, reddish-brown scum forms at the site. That is the clot or coagulum. Physically, what is it? It is a three-dimensional net. It's a dense network of protein threads called fibrins. Inside this sticky net, dead and damage-formed elements of red blood cells and platelets get physically trapped, creating a biological plug.
But earlier we established that those fibrin threads don't just exist normally in the blood, because if they did, our blood would constantly be full of sticky nets and would turn into jelly inside our veins. We would instantly have strokes and heart attacks.
And that introduces the genius of the biological cascade. A cascade is a sequence of precise triggers, where one enzyme activates another, which activates another. It exists as a safety mechanism. Fibrin threads don't exist in healthy blood. They exist as that inactive soluble plasma protein we talked about earlier, fibrinogen.
So fibrinogen is the inactive folded up net. Fibrin is the active diploid net. How does it convert?
It requires a specific enzyme to cleave it and open it up. That enzyme is called thrombin. Thrombin cuts the fibrinogen, exposing sticky ends, causing them to link together into long fibrin threads.
But wait, the same safety problem applies. Thrombin can't just be floating around active all the time, or it would constantly trigger the fibrinogen.
Exactly. You're seeing the layered safety protocols. Thrombin itself doesn't normally exist. It circulates as an inactive precursor called prothrombin.
Okay, so what turns inactive prothrombin into active thrombin?
An enzyme complex called thrombokinase. And thrombokinase is only formed when there's an injury. Let's trace the dominoes falling in real time.
All right, so I slice my finger on a piece of paper. Trauma occurs.
That trauma rips open endothelial cells and exposes collagen. The platelets flowing by hit that rough collagen and essentially explode. Yeah. They stimulate the release of specific factors. The damaged tissue itself also releases factors. This localized release of alarm chemicals initiates a massive localized cascade involving nearly a dozen different factors in the plasma that are normally turned off.
Domino A hits B, which hits C, which forms the thrombokinase complex.
Yes. Thrombokinase then acts on the inactive prothrombin, converting it to active thrombin. The newly minted thrombin acts on the inactive fibrinogen, converting it into active fibrin threads. The threads weave together, trap the red blood cells, and the hole is plugged.
It's an elegant, localized amplification. One injured cell triggers 100 platelets, which trigger 1,000 thrombin molecules, which trigger 1,000,000 fibrin threads. And here is what you must remember for this specific process. There is a linchpin, a specific mineral ion that is absolutely essential for almost every step of this cascade to function.
Calcium. Calcium ions play a vitally important role in clotting. In fact, when blood is drawn for donation, the blood bank bags contain chemicals like citrate or oxalate. Why? Because those chemicals bind up and remove all the calcium from the blood. Without free calcium, the cascade is completely paralyzed, and the donated blood stays liquid in the bag for weeks.
Wow. So a severe calcium deficiency doesn't just affect your bones, it fundamentally compromises your ability to stop bleeding. Okay, so we know how the blood flows and we know how it patches its own leaks. But what happens if the leak is too big? Someone loses massive amounts of blood, they are rushed to the hospital, and they need a transfusion. We've all heard of blood typing, A, B, O, positive, negative. But why is it so strictly regulated? Why can't I just take human blood from anyone? It all looks the same.
It looks the same macroscopically, but immunologically it is vastly different. It all comes down to the immune system's primary directive. Identify self versus non-self. The body is constantly scanning for invaders. The two most widely utilized grouping systems globally are the ABO grouping and the RH grouping.
Let's unpack the ABO system first. It's essentially a system of microscopic ID badges and security bouncers.
Exactly. The ID badges are called antigens. Specifically, these are complex sugar structures projecting outward from the surface of your red blood cells. The ABO system recognizes the presence or absence of two distinct antigens, antigen A and antigen B.
And the security bouncers are the natural antibodies floating in your blood plasma. These are proteins pre-programmed to latch onto and destroy any ID badge that doesn't belong in your body.
Let's run through the combinations.
Yeah.
If you have blood group A, it means your red blood cells proudly display the A antigen ID badge. Because you are A, your immune system knows A is safe. But it views B as an alien threat. Therefore, your plasma naturally contains anti-AB antibodies.
So if a nurse accidentally gives you group B blood, those anti-B bouncers will instantly recognize the foreign B antigens, latch onto the donated red blood cells, and cause them to clump together, a process called agglutination, and destroy them, which can be fatal.
Correct. The reverse is also true. If you are blood group B, your cells have the B badge, and your plasma is armed with anti-antibodies.
What happens if a person inherits both badges? Blood group AB.
If you are blood group AB, your red blood cells have both the A and B antigens. Now think about the security system. If your plasma contained anti-A or anti-B antibodies, your own immune system would attack your own blood. So by sheer necessity, individuals with group AB blood have NO natural antibodies against A or B in their plasma.
And because they have no bouncers in their plasma to attack incoming blood, they can accept blood from group A, group B, group AB, or group O. That is the physiological reason why people with AB blood are known as universal recipients.
Spot on. Now look at the total opposite scenario. Blood group O. The O essentially stands for 0. These individuals have NOA or B antigens on the surface of their red blood cells. Their cells are blank. They have no ID badges.
Because they have no ID badges, they are basically invisible to the security bouncers of any other blood type. You can inject group O red blood cells into an AB or AB person, and their immune system won't react because there's nothing for the antibodies to grab onto. Hence, group O individuals are the universal donors.
But the catch for the Group O individual is that because their body has never seen an A or B antigen, their plasma is heavily armed with both anti-A and anti-B antibodies. A Group O person can give to anyone, but can only safely receive blood from another Group O person.
That matrix of antigens and antibodies dictates the rules of all emergency medicine. But the ABO system is only the first layer. The other half is the RH grouping. And this is where the biology gets incredibly dramatic, specifically regarding pregnancy. The RH trap.
This is one of the most fascinating physiological traps in human medicine. The Rh antigen gets its name because it was first identified in rhesus monkeys. It's another protein ID badge on the surface of the RBC. It is present in nearly 80% of humans.
If you have it, you are Rh positive. If you lack it, you are Rh negative. That's what the positive or negative means when someone says they are A positive or O negative.
Right. And just like the ABO system, if you take Rh positive blood and put it into an Rh negative person, the Rh negative immune system will see that Rh antigen as a foreign invader and start building specific anti-Rh antibodies to destroy it. Therefore, Rh groups must be matched before transfusions. But the truly critical scenario is a condition called erythroblastosis fetalis.
Let's paint the picture. You have a very specific, common genetic pairing. An Rh negative mother gets pregnant, and the fetus inherits the Rh positive gene from the father. So Rh negative mother, Rh positive baby.
During the first pregnancy, there is almost never an issue. Why? Because the placenta is a remarkably effective barrier. It allows nutrients and oxygen to cross, but it keeps the maternal blood cells and the fetal blood cells completely separated. The mother's immune system remains totally blind to the fact that the fetus has Rh positive blood. The first baby is born perfectly healthy.
But the trap is set during delivery.
Yes. The process of birth is traumatic to the tissues. As the placenta detaches from the uterine wall, microscopic tears occur. There's almost always a small mixing of blood. A tiny volume of the baby's Rh positive blood spills into the mother's Rh negative bloodstream.
And suddenly the mother's immune system sees the invader.
It sees the Rh antigen. The baby's already born and perfectly safe. But inside the mother, her white blood cells sound the alarm. They identify the foreign Rh protein and begin the slow process of manufacturing specialized anti-Rh antibodies. More importantly, her immune system creates memory B cells. She is now permanently sensitized. Her body will remember the Rh antigen forever.
And this is where the danger lies for subsequent pregnancies. What happens if she gets pregnant a second time with another Rh positive fetus?
This is the tragedy of erythroblastosis fetalis. In the second pregnancy, the mother's immune system is already primed and armed. And here's the lethal biological quirk. While entire red blood cells cannot cross the placenta, the specific anti-Rh antibodies the mother produced are small enough molecules, specifically IgG type, to slip right through the placental barrier.
Oh, wow. So the mother's antibodies leak into the developing fetus's bloodstream. And because the fetus is Rh positive, those antibodies start relentlessly attacking and destroying the baby's red blood cells while it's still in the womb.
It is devastating. It causes massive, severe anemia in the fetus. The fetus's liver and spleens go into overdrive, trying to produce new red blood cells, which leads to severe jaundice upon birth, or it can be fatal to the fetus in utero.
But the incredible thing is that modern medicine has completely solved this. It's an elegant intervention. How do we stop the mother's immune system from setting the trap?
We trick it. We can entirely avoid this sensitization by administering a shot of synthetic anti-Rh antibodies, often known by the brand name RhoGAM, to the Rh negative mother immediately after the delivery of her first Rh positive child, usually within 72 hours.
Wait, I need to make sure I understand this. The problem is the mother making antibodies. So our solution is to inject her with antibodies. How does that not hurt her?
Because of timing. When the baby's Rh positive cells spill into the mother's blood during birth, it takes her immune system days or weeks to recognize them, process them, and build a permanent memory of them. When we inject the RhoGAM antibodies right after birth, those synthetic antibodies aggressively hunt down and destroy any stray fetal Rh positive cells floating in the mother's blood before the mother's own immune system even has a chance to notice they are there.
That is mind-blowing. We are basically sending in a cleanup crew to mop up the evidence. The synthetic antibodies destroy the fetal cells, and then the synthetic antibodies naturally degrade over a few weeks. The mother's immune system stays completely naive, She never forms memory cells, meaning her next pregnancy will be just as safe as her first.
Exactly. It's one of the great triumphs of preventative immunology. Now, before we leave the fluids entirely and move to the mechanics of the heart pump, we have to talk about the middleman, the shadow network I mentioned earlier, lymph.
Right. We said blood carries the groceries to the cells, but blood vessels are closed tubes. It's a closed plumbing system. The red blood cells don't actually leave the blood vessels to visit the muscle cells. So how do the oxygen and the glucose actually get from inside the closed pipe to the living cells that surround it?
Pressure and permeability. As blood is pumped away from the heart and reaches the tissues, it enters the capillaries. Capillaries are the smallest blood vessels, and their walls are only one single cell thick. Furthermore, they have microscopic pores. Because the blood is under hydrostatic pressure from the beating heart water, and small water-soluble substances like glucose, amino acids, and oxygen are physically squeezed out through the capillary pores into the empty spaces between the tissue cells.
It's like a soaker hose in a garden. The water bleeds out through the fabric, but the larger stuff stays inside.
Correct. The large plasma proteins like the fibrinogens and albumins we talked about, and all the formed elements, the red and white blood cells, are simply too massive to fit through the pores. They stay inside the blood vessel.
So this clear, nutrient-rich fluid that gets squeezed out bathing every single cell in your body is called interstitial fluid or tissue fluid.
And here is what you must remember. This tissue fluid has the exact same mineral distribution as plasma. It is the literal medium of exchange. The blood never directly touches your muscle cells. Oxygen diffuses from the blood into the interstitial fluid and then into the cell. The cell dumps its carbon dioxide into the interstitial fluid, which then diffuses back into the blood. It is the vital middleman of all biological exchange.
But if this fluid is constantly, relentlessly being squeezed out of the capillaries by blood pressure, wouldn't our tissues just fill up with water? We would swell up and burst.
We would if we didn't have a secondary drainage system, and that is the lymphatic system. It is an elaborate parallel network of blind-ended vessels that snake through all your tissues. These vessels act as drains, collecting all this excess interstitial fluid. They slowly funnel it through larger and larger lymphatic vessels, passing it through lymph nodes for security checks, and eventually dump it back into the major veins near your collarbones, returning the fluid to the blood circulation.
And once that interstitial fluid crosses into the lymphatic vessels, it gets a name change. It is officially called lymph.
Exactly. Lymph is a colorless fluid, but it isn't just passive drainage water. It has two incredibly unique high-yield functions that you need to know. First, it is the primary staging ground for the immune system. The lymph nodes are packed with highly specialized lymphocytes, those T cells and B cells we discussed. As the lymph fluid slowly drains through the nodes, the lymphocytes inspect it for bacteria or viruses. If they find an invader, they trigger a massive immune response. That's why your lymph nodes swell up when you are sick.
They are literal security checkpoints. And the second unique function is related to digestion, which seems totally out of place.
It does, but it's a structural necessity.
Mm.
Here's the key concept. Lymph is the specific exclusive pathway for fat absorption. When you eat carbohydrates or proteins, they are broken down into small water-soluble molecules that get absorbed directly into the blood capillaries in your intestines. But fats are different.
Fats don't dissolve in water. Oil and water don't mix.
Exactly. Digested fats assemble into large complexes that are simply too big to squeeze into the tiny blood capillaries. So inside the villi, the tiny finger-like projections lining your small intestine there are specialized, highly permeable lymphatic capillaries called lacteals. The large fat molecules can easily pass into the lacteals, travel through the lymphatic system, and eventually get dumped into the bloodstream much higher up in the chest.
Fats bypass the initial bloodstream and take the lymph highway. That perfectly wraps up our understanding of the fluids. The blood river, the middleman tissue fluid, and the lymphatic drainage network. Now we need to shift gears. A river doesn't flow under pressure without a pump. So let's look at the evolutionary blueprint and heart anatomy.
Let's zoom out again to evolution. We established that complex animals need internal transport, but nature experimented with different plumbing blueprints. Broadly, we classify them as open circulatory systems and closed circulatory systems.
Arthropods, which are your insects, spiders, crabs, and mollusks like snails and clams, they operate on an open circulatory system. They have a heart that pumps blood through some large vessels, but then the vessels just end. The blood is dumped directly into wide open spaces or body cavities called sinuses. The internal organs literally just bathe in a pool of blood.
It works for them, but it is low pressure and sluggish. Compare that to annelids like earthworms and chordates, the group that includes us vertebrates. We evolved a closed circulatory system. The blood pumped by our heart is entirely confined within a closed continuous network of blood vessels. It never just sloshes around freely in the body cavity.
It's the difference between a swamp and a pressurized municipal water grid. Why is the closed pattern considered more advantageous?
Control and speed. Because the fluid is confined to pipes, you can generate high pressure, which means the blood travels much faster, delivering oxygen more rapidly to support higher metabolisms. Furthermore, a closed system allows for precise regulation. If you start running, your body can actively constrict the blood vessels going to your stomach and dilate the vessels going to your leg muscles, dynamically rerouting the blood flow to where it's needed most. You can't redirect fluid in an open bathtub.
So vertebrates committed to the closed system driven by a muscular chambered heart. But the heart itself went through a massive evolutionary upgrade process to support higher life forms. Let's trace that evolution because understanding the fish heart explains exactly why the human heart is built the way it is.
That's a beautiful progression. Let's look at fishes. Fishes possess the most basic vertebrate heart, a two-chambered pump. It consists of one atrium, which is a receiving chamber, and one ventricle, which is the heavily muscled pumping chamber.
And the most counterintuitive, fascinating thing about the fish heart is that it only ever handles deoxygenated blood. It never sees oxygen.
Right, it's what we call single circulation. The heart's ventricle forcefully pumps oxygen-poor dark blue blood forward to the gills. As the blood passes through the delicate capillaries of the gills, it picks up oxygen from the water and becomes bright red. But from the gills, it does not go back to the heart. It flows directly onward to the rest of the body tissues.
The body cells consume the oxygen, turn the blood blue again, and that deoxygenated blood lazily makes its way back to the atrium of the heart to complete the single loop. Heart to gills to body to heart.
It works in water, but because the blood has to squeeze through the tiny capillaries of the gills before reaching the body, it loses almost all of its pressure. It's a very low pressure system. To live on land fighting gravity, we needed higher pressure. Moving up the evolutionary ladder, amphibians and reptiles, with the notable exception of crocodiles, evolved a three-chambered heart, two atria and one single ventricle.
This introduces a second loop, incomplete double circulation. The left atrium receives fresh oxygenated blood returning from the lungs or skin. The right atrium receives the deoxygenated blood returning from the body tissues. They are separate at the top. But then both the left and right atria dump their contents down into the exact same single ventricle.
Which creates an obvious mechanical flaw. The highly oxygenated blood and the deoxygenated waste blood physically mix together in that single ventricle before being pumped back out to the body. It is incredibly inefficient. The body receives a diluted mixture of oxygen.
It's like piping your fresh spring water and your sewer water into the same holding tank before pumping it to your kitchen sink. Why would nature settle for that?
Because amphibians and reptiles are cold-blooded. They have much lower metabolic demands than we do. They don't need to burn massive amounts of oxygen just to keep their body temperature at 98 degrees. The mixed blood is good enough for their lifestyle. But for warm-blooded animals, endotherms, who have sky-high metabolic rates, that mixing is a deal breaker, which leads us to crocodiles, birds, and mammals. We possess the peak of this design, a four-chambered heart, two atria, two ventricles.
True double circulation. Complete separation. The oxygenated blood stays strictly on the left side of the heart, and the deoxygenated blood stays strictly on the right side. The ventricles pump them out into two completely isolated parallel pathways.
Exactly. So let's zoom in and focus entirely on the anatomy of the human heart. Location first. It's situated right in the middle of the thoracic cavity nested between the two lungs, but its apex is slightly tilted toward the left, which is why we feel our heartbeat on the left side of our chest.
It's roughly the size of a clenched fist. Embryologically, it is derived from the mesoderm germ layer. And it's not just sitting bare in the chest, it is heavily protected by a double-walled membranous bag called the pericardium.
The pericardium is brilliant. It's like a tough balloon enclosing another balloon, and the space between the two walls is filled with a slippery, lubricating fluid called pericardial fluid. The heart is a muscle that physically expands and violently contracts 100,000 times a day. If it were rubbing against the lungs and ribs, the friction would destroy it. The pericardium allows it to beat completely friction-free.
Okay, let's open up the engine block and look at the chambers. We have the four rooms. The two relatively small, thin-walled upper chambers are the atria. The two massive, thick-walled lower chambers are the ventricles. To ensure that complete separation of blood we just talked about, we have solid muscular walls called septa dividing the rooms.
Let's map the septa. The wall separating the right and left atria is called the interatrial septum. It's relatively thin because the atria don't generate much pressure. But the wall separating the left and right ventricles, the interventricular septum, is incredibly thick and heavily muscled.
And there's a thickness difference between the left and right ventricle walls themselves, right? The left ventricle is the thickest muscle in the entire heart.
A fantastic observation, and it's pure physics. The right ventricle only has to pump blood right next door to the lungs. It's a short, low-resistance trip. The left ventricle, however, has to generate enough force to pump blood up to your brain against gravity and all the way down to your toes and push it through miles of tiny capillaries. It requires massive force, so the muscle hypertrophy gets incredibly thick. Finally, separating the upper atria from the lower ventricles is a thick plate of fibrous tissue called the atrioventricular septum.
But those horizontal septa can't be solid walls. If they were, the blood couldn't flow from the top rooms to the bottom rooms. They must have doorways. And those doorways are guarded by the most critical mechanical components of the heart, the valves.
This is a massive focus area for understanding hemodynamics and a classic spot where medical students and biology learners flip the facts. You need to memorize which valve is on which side of the heart.
Let's map it. The right side first.
The opening between the right atrium and the right ventricle is guarded by a large valve composed of 3 distinct muscular flaps or cusps. Therefore, it is called the tricuspid valve.
Tricuspid on the right and the left side.
The opening between the left atrium and the left ventricle is guarded by a valve with only 2 flaps. It is the bicuspid valve. In clinical medicine, it is almost exclusively referred to as the mitral valve because the 2 flaps resemble a bishop's mitre, a tall 2-peaked hat.
So tricuspid on the right, bicuspid, or mitral on the left. And there are 2 more valves at the primary exits of the heart.
Yes. When the right ventricle squeezes to pump blood out into the pulmonary artery toward the lungs, and when the left ventricle squeezes to pump blood out into the massive aorta toward the body, those main exit doors are guarded by the semilunar valves. They are named because their flaps look like little half-moons.
And what is the singular, non-negotiable job of every single one of these valves?
To ensure strict one-way traffic, blood must move from the atria down to the ventricles and from the ventricles out to the arteries. When the powerful ventricles contract, the immense pressure threatens to blast the blood backward into the atria. The tricuspid and bicuspid valves catch that pressure and violently snap shut, sealing the pathway and preventing any backward regurgitation.
It is a mechanical masterpiece. But a pump, no matter how good the valves are, needs an engine. It needs a spark to tell it when to squeeze. This brings us to the nodal tissue, the heart's internal electrical grid.
This is one of the most profound physiological truths. We often assume the brain controls everything. But if you were to carefully sever all the nerves leading from the brain to a living human heart, the heart would not stop. It would continue to beat rhythmically entirely on its own.
That is wild. The heart is completely self-reliant.
Biologically, we say the heart is autoexcitable or myogenic, while the entire heart is made of specialized cardiac muscle. A specific subset of these muscle fibers has evolved to act like electrical wiring. This is the nodal tissue. These tissues don't contract very well. Instead, their job is to generate and conduct electrical action potential sparks without any external stimuli.
Let's trace the wiring. Where does the spark actually originate?
The primary generator is a small patch of tissue located in the upper right corner of the right atrium. This is the sinoatrial node, the ASN. It is unique because it can spontaneously generate electrical impulses faster than any other tissue in the heart, roughly 70 to 75 action potentials per minute.
And because it fires the fastest, it forces the entire rest of the heart muscle to dance to its rhythm. If the SAN fires 72 times a minute, the heart beats 72 times a minute. That is why the SAN is universally known as the natural pacemaker of the heart.
Precisely. So the SAN fires. The electrical wave sweeps rapidly across the walls of both the left and right atria, causing them to physically contract. But the electricity doesn't just flow smoothly down into the ventricles. There is a layer of fibrous non-conducting tissue separating the top and bottom of the heart. The electrical signal can only cross through one specific gateway.
And that gateway is the second nodal mass, the atrioventricular node, or AVN, located in the lower left corner of the right atrium, right near the septum.
Here's a fascinating detail that highlights the genius of the system. The AVN acts as a relay station, but it deliberately delays the electrical signal by about 1/10 of a second before sending it down.
Wait, why would it want to slow the signal down? Wouldn't you want the heart to contract as fast as possible?
Think about the fluid dynamics. The atria are squeezing blood down into the ventricles. If the electrical signal went straight from the top of the heart to the bottom, Instantly, the atria and the ventricles would contract at the exact same time. They would be squeezing against each other. By delaying the signal for zero point 0.1 seconds, the AV node gives the atria enough time to finish emptying their blood into the ventricles before the massive ventricles begin their violent contraction.
That is just incredible biological engineering. The delay is the secret to the pump's efficiency. Okay, so after that brief delay at the AV node, where does the spark go?
The signal travels down a specialized bundle of fibers called the atrioventricular bundle, or the AV bundle of His. This bundle punches through the fibrous septum and runs straight down the top of the interventricular septum, the thick wall separating the two ventricles.
And right there, it splits. It divides into a right bundle and a left bundle, routing the electricity to both sides of the heart.
From those bundles, a vast network of microscopic, highly conductive fibers branches out and penetrates deep into the massive muscular walls of the ventricles. These terminal branches are called Purkinje fibers. They deliver the electrical shock simultaneously to all the ventricular muscle cells, causing the massive upward squeeze that ejects the blood from the heart.
SN to AVN, delayed on the bundle of His splitting left and right and exploding outward through the Purkinje fibers. That electrical choreography is what directly causes the physical squeezing of the blood, which leads us into exactly how that 0.8 seconds plays out.
The 0.8 second miracle, the cardiac cycle.
Let's do the math on this. A normal resting heart rate is 72 beats per minute. 60 seconds divided by 72 beats gives you exactly 0.83 seconds. A complete sequence of events, one heartbeat takes 0.8 of a second. Let's slow time down and walk through exactly what is happening inside the chambers during that fraction of a second.
Let's start from a moment of complete rest. Imagine all 4 chambers of the heart are totally relaxed. In biology, muscle relaxation is called diastole. So we are in joint diastole.
In this relaxed state, the pressure inside the heart is low. The heavy tricuspid and bicuspid valves are hanging open. Oxygenated blood from the lungs is pouring into the left atrium, and deoxygenated blood from the body is pouring into the right atrium. And because those valves are open, the blood just falls right through the atria and passively begins filling up the relaxed ventricles below. The exit doors, the semilunar valves, are tightly closed.
Then the sequence begins. The pacemaker, the SAN, fires its electrical spark. This causes both the left and right atria to actively contract simultaneously. Muscle contraction is called systole, so we enter atrial systole.
The atria squeeze. Now, because the ventricles were already passively filling up while the heart was resting, they are mostly full. This atrial squeeze just acts as a final top-off. It pushes the remaining 30% of the blood down into the ventricles, stretching them to their maximum capacity.
Meanwhile, that electrical spark has hit the AV node, waited its 0.1 seconds, and is now racing down the Purkinje fibers into the massive ventricular walls.
And the main event happens. Ventricular systole, the massive lower chambers begin to violently contract. At the exact same moment, the atria relax, going back into diastole to start receiving blood again.
Now visualize the pressure mechanics. The powerful ventricles are rapidly squeezing a non-compressible fluid. The pressure inside the ventricles spikes exponentially in milliseconds. The blood is looking for an escape route, and it immediately tries to surge backward up into the low-pressure atria.
But the tricuspid and bicuspid valves are standing in the way. The backward surge of blood catches the flaps of those valves like a parachute and slams them shut. And that physical slamming together of the valve cusps is the very first heart sound you hear when you listen through a stethoscope. Lub.
Exactly. The lub, or the first heart sound S1, is specifically the closure of the atrioventricular valves. Now the valves are shut, but the ventricles are still squeezing. The pressure keeps rising higher and higher until it eventually overcomes the pressure of the blood sitting out in the arteries.
The pressure forces the exit doors, the semilunar valves, to blow open. The blood forcefully rushes out of the right ventricle into the lungs and out of the left ventricle into the aorta to feed the body. The contraction finishes. The ventricles have emptied their stroke. Now they relax. Ventricular diastole.
As the thick ventricular muscle relaxes, the space inside expands and the pressure inside the ventricles plummets. Now the situation is reversed. The high-pressure blood that was just ejected into the aorta and pulmonary artery tries to flow backward down the pressure gradient back into the relaxing ventricles.
But again, the valves catch it. The backward-flowing blood fills the cup-like flaps of the semilunar valves, snapping them tightly shut. And that second slamming of doors is the second heart sound. Dub.
The dub, or the second heart sound S2, is the closure of the semilunar valves. If a doctor hears a whoosh or a hiss between the lub and the dub, that's a heart murmur. It means one of those valves isn't sealing perfectly and blood is leaking backward. But in a healthy heart after the dub, the ventricular pressure continues to fall until it is lower than the pressure in the atria above them. The weight of the blood waiting in the atria pushes the tricuspid and bicuspid valves open again. Blood flows passively into the ventricles, we're back in joint diastole, and the entire cycle is ready to start over, all in zero point 0.8 seconds.
Lub-dub pause, it is mesmerizing fluid mechanics. And the numbers behind it are just as impressive. We can calculate the exact volume of this biological pump.
This is a high-yield concept for understanding cardiovascular health. During each single cardiac cycle, one ventricle pumps out a specific volume of blood. This is called the stroke volume. And in a healthy resting adult, it is approximately 70 milliliters.
So stroke volume is 70 ml per beat, roughly a quarter cup of liquid.
Now, to find out the total work the heart is doing, you calculate the cardiac output. The formula is simple. Cardiac output equals the stroke volume multiplied by the heart rate.
So 70 ml per beat multiplied by an average of 72 beats per minute. That comes out to roughly 5,040 milliliters, or about 5 liters.
Exactly 5 liters per minute, which happens to be the total volume of blood in your body. It means your entire blood supply cycles through your heart once every single minute while you were just sitting still.
And the heart is dynamic. If I start sprinting, my cells need more oxygen. My heart rate goes up to 150 beats per minute, which obviously increases the cardiac output. But the stroke volume can change too, right?
Absolutely. The body can alter both variables. The sympathetic nervous system makes the heart muscle contract more forcefully, squeezing out more than 70 ml per beat. This is why an elite marathon runner might have a resting heart rate of only 40 beats per minute. Their heart muscle has hypertrophied. It's so strong and efficient that their stroke volume is massive. They can maintain that 5 liter per minute cardiac output with far fewer beats.
Okay, we just described the physical squeezing and the fluid dynamics. Now I want to connect that to the electricity we discussed earlier, and specifically to the most famous medical machine in pop culture, the monitor you see on every hospital TV show with the squiggly green line.
The electrocardiograph.
Oh.
And here's a pedantic but important terminology trap that catches a lot of students. The machine itself, the box with the wires, is the electrocardiograph. The graphical output, the actual squiggly line printed on the paper or displayed on the screen, is the electrocardiogram or ECG. Graph is the machine. Gram is the picture.
A standard setup for an ECG involves placing electrical leads on the patient. To get a basic reading, you use a triangle configuration. One lead on the right wrist, one on the left wrist, and one on the left ankle. This continuously monitors the electrical activity sweeping through the heart.
For a detailed clinical evaluation, cardiologists attach multiple leads across the chest to view the heart in 3D. But the fundamental squiggly line, the standard ECG trace, is what we need to decode. It is a direct visual representation of that electrical pathway we traced from the SAN down to the Purkinje fibers.
We need to translate those specific squiggly lines, the peaks and valleys, into the physical heart actions. Each major wave is identified with a letter from P to T. Let's start with the first small bump.
The P wave. The P wave represents the electrical excitation or depolarization of the atria.
Let's clarify depolarization really quickly so it's not just jargon. In a resting state, muscle cells have a negative electrical charge inside compared to the outside. When the signal hits them, specific ion channels open, sodium rushes in, and the cell's charge suddenly flips to positive. That electrical flip is depolarization.
Perfect explanation. That electrical flip, the P wave, is the spark that sweeps across the atria. Importantly, this electrical wave is what leads directly to the physical contraction. The P wave happens, and milliseconds later, the atria physically squeeze.
P wave equals atrial depolarization. Then the trace drops back to baseline for a split second, which perfectly aligns with that 0.1 second delay at the AV node we talked about. Then you get a slight downward dip, followed by a massive, sharp, towering upward spike. This is the QRS complex.
The QRS complex represents the depolarization of the ventricles. This is the massive electrical signal racing down the bundle of His and exploding outward through the Purkinje fibers. It represents the electrical trigger for the main event.
Because the ventricles are so heavily muscled and contain so many cells, their electrical signal is vastly larger than the atria's, hence the massive spike on the graph. The physical contraction of the ventricle's ventricular systole actually begins shortly after the Q-dip and continues through the complex.
And here is a highly practical clinical use of the QRS complex. Because each QRS spike represents exactly one ventricular contraction, one heartbeat, you can simply count the number of QRS complexes that occur within a specific time period on the ECG paper to instantly determine a patient's heart rate.
Count the spikes, get the pulse. Finally, after the QRS complex, the line returns to baseline, and then there's a third, broader, softer bump, the T wave.
The T wave represents the ventricles resetting. The biological term is repolarization. The cells are pumping the ions back to their original positions, returning from an excited state back to a normal resting state. The end of the T wave marks the physical end of ventricular systole.
Wait, if the T wave is the ventricles resetting, where is the wave for the atria resetting? They had to reset too, right?
They absolutely do. The atria repolarize at the exact same time the ventricles are depolarizing. But because the electrical signal of the massive ventricles, the QRS complex, is so huge and powerful, it completely overshadows and masks the tiny electrical signal of the atria resetting. You simply can't see it on the graph.
That makes perfect sense. Here is what you must remember for the ECG. P is atria excitation, QRS is ventricle excitation, T is ventricle reset. Why is this so crucial in medicine?
Because the ECG shapes for a healthy heart are universally standardized. The timing intervals between the waves are precise to the millisecond. Any deviation, like a flattened T wave, an elongated gap between the P and the Q, an inverted QRS, tells the doctor exactly where the electrical pathway is broken or exactly which part of the heart muscle is dying from lack of oxygen. It is an unparalleled, non-invasive diagnostic tool.
All right, we understand the fluids, we understand the pump, and we understand the electrical grid. Now let's trace the actual roadmap, the blood vessels, and the mechanics of double circulation.
The blood flows strictly by a fixed route through a vast network of arteries and veins. And regardless of whether it's an artery carrying blood away from the heart or a vein carrying blood back, the wall of the vessel essentially consists of three distinct layers.
The innermost layer, the part the blood actually touches, is the tunica intima. It's made of squamous endothelium.
And squamous just means flat and scale-like. The cells are arranged like perfectly smooth floor tiles. This provides an utterly frictionless surface for the blood cells to glide over without sticking or tearing.
Outside of that frictionless inner tube is the middle layer, the tunica media. This layer is made of smooth muscle and elastic fibers. This is the layer that can contract or relax to change the diameter of the blood vessel. And finally, wrapping the whole pipe is a tough external layer of fibrous connective tissue with collagen called the tunica externa, which provides structural integrity.
Now here is a major focal point for anyone studying cardiovascular anatomy. Arteries and veins have very different jobs, so their structure differs. Arteries carry blood away from the heart immediately after the ventricle squeezes. They have to withstand the massive violent pressure pulses of the heartbeat.
So they need thick walls to absorb that shock without bursting.
Exactly. Conversely, veins carry blood back to the heart. By the time the blood has gone through the capillary beds, almost all the pressure from the heart's pumping is gone. Veins are low-pressure vessels. Therefore, the key anatomical difference you must know is that the tunica media, that muscular elastic middle layer, is comparatively much thinner in veins than in arteries.
Because the veins are low pressure, they don't have the force of the heart to push the blood back up your legs against gravity. How does the blood get back?
Veins rely on two hacks. First, your skeletal muscles. When you walk your calf muscles, squeeze the veins, physically milking the blood upward. Second, veins have internal one-way valves. When the muscle squeezes, blood moves up. When the muscle relaxes, the valve snaps shut so the blood doesn't fall back down to your ankles. Arteries don't need these valves because their high pressure keeps blood moving in one direction.
Brilliant. Now let's trace the full map of our four-chambered heart, the double circulation. We have two distinct loops running simultaneously.
Loop number one is the pulmonary circulation. This is the short loop, purely dedicated to gas exchange. It starts in the right ventricle, which pumps dark deoxygenated blood into the pulmonary artery.
Wait, I thought arteries always carry oxygenated blood.
That is the most common physiological trap. Arteries simply carry blood away from the heart. Veins carry blood to the heart. The pulmonary artery is carrying blood away from the heart to the lungs, so it's an artery, but it is carrying oxygen, poor blood.
Ah, right. So the pulmonary artery takes the blue blood to the lungs. In the lung capillaries, the blood dumps its CO2 and picks up fresh oxygen. The newly bright red oxygenated blood is then carried back to the left atrium of the heart via the pulmonary veins. That is the complete pulmonary circuit. Heart to lungs, back to heart.
Now for the masses, loop number two. Systemic circulation. This is the delivery network. It starts in the left ventricle, which forcefully pumps the highly oxygenated blood out into the body's main arterial highway, the aorta.
The aorta branches into a sprawling network of arteries, then smaller arterioles, and eventually the microscopic capillaries woven through every single tissue in the body. The systemic circulation provides the O2 and nutrients to the cells and picks up the CO2 and waste.
That deoxygenated blood is then collected by a system of small venules, larger veins, and finally the massive vena cava, which empties it all back into the right atrium to start the whole process over again. That is the systemic circuit. Double circulation simply means the blood passes through the heart twice for every one complete trip around the body.
But within that systemic loop, there are two specialized subpathways we need to highlight.
Yes. The first is a detour called the hepatic portal system. Normally, blood goes from the heart to an artery, into a capillary bed in an organ, into a vein, and straight back to the heart. But the hepatic portal system inserts a second capillary bed into the sequence. It's a unique vascular connection between your digestive tract and your liver.
When you absorb nutrients and potentially toxins or alcohol from your intestines, that blood doesn't go back to the heart right away to get pumped to the rest of the body. The hepatic portal vein carries that nutrient-rich, unfiltered blood straight into the liver first.
Exactly. It's a screening mechanism. It gives the liver the first pass to process the nutrients, store the excess glucose as glycogen, and detoxify poisons before that blood is allowed into the general systemic circulation. It protects the brain and heart from absorbing toxins directly from the gut.
And the second special pathway is arguably the most critical for survival, the coronary system.
Think about the irony of the heart. The heart is constantly filled with gallons of fresh oxygenated blood. But the cardiac muscle tissue is so thick that the oxygen in the chambers cannot simply diffuse outward to feed the muscle cells. The heart would starve while full of food.
So the very first branches off the aorta, right at the base where it leaves the left ventricle, are the coronary arteries. They immediately wrap back around the outside of the heart, diving into the muscle tissue. The heart has its own dedicated private system of blood vessels exclusively for the circulation of blood to and from the cardiac musculature. It is the absolute definition of secure your own oxygen mask before assisting others.
Speaking of regulating the heart, we know the heart is myogenic. It generates its own beat. But it still needs a central command to adjust its speed based on the body's needs. If a tiger jumps out at you, your heart rate needs to spike instantly.
The central command center for this is a special neural hub located in the brainstem, specifically the medulla oblongata. It monitors blood pressure and oxygen levels, and it can moderate cardiac function through the autonomic nervous system, or ANS.
The ANS has two distinct branches that act exactly like a gas pedal and a brake on the heart's natural pacemaker. Neural signals sent through the sympathetic nerves act as the gas pedal. If the medulla senses danger or exercise, sympathetic nerves release neurotransmitters that increase the rate of the heartbeat, increase the physical strength of the ventricular contraction, and therefore massively increase cardiac output.
On the flip side, when the danger is gone and you sit down to rest, the parasympathetic neural signals kick in. They act as the brake. They decrease the heart rate, slow down the speed of conduction of the action potential through the AV node, and decrease the cardiac output, bringing you back to a resting baseline.
And it's not entirely neural. The endocrine system plays a role too.
Yeah.
Your adrenal glands sitting on top of your kidneys release adrenal medullary hormones like adrenaline and noradrenaline. When these hormones hit the bloodstream, they act directly on the heart muscle to independently increase cardiac output during a fight or flight response.
Okay, we have built an incredible, intricate understanding of how this machinery works perfectly. But we also need to look at the reality of human health. What happens when the plumbing gets compromised? Let's cover the disorders of the circulatory system.
The most prevalent one globally is hypertension, more commonly known as high blood pressure. When a doctor wraps that cuff around your arm, they are measuring the pressure inside your systemic arteries. A normal healthy reading is considered 120 over 80 millimeters of mercury.
We talked about systole and diastole earlier. So the higher number, the 120, is the systolic pressure, the maximum force against the artery walls when the left ventricle is actively contracting. The lower number, the 80, is the diastolic pressure, the resting pressure in the arteries while the ventricle is relaxing and refilling.
Correct. If an individual has repeated checks and their blood pressure is consistently reading 140 over 90 or higher, they are clinically diagnosed with hypertension. And this isn't just a number on a chart. It is a mechanical crisis.
Right, think of a garden hose. If you crank the water pressure to the absolute maximum and leave it there for years, the hose degrades. The constant high-pressure shear force physically damages the delicate squamous endothelium lining the arteries. It leads to microscopic tears.
And the damage compounds. It drastically increases the workload on the heart. The left ventricle has to push much harder to open the aortic valve against that high back pressure. Over time, hypertension relentlessly damages vital end organs, particularly the delicate capillary beds in the brain, increasing the risk of stroke, and the kidneys leading to renal failure. It is often called the silent killer because you can't feel high blood pressure until something breaks.
Which leads directly into the second major disorder, coronary artery disease, or CAD, often referred to colloquially as atherosclerosis. We just established that the heart relies entirely on those tiny coronary arteries wrapping its exterior to supply its own oxygen. CAD specifically targets these vital supply lines.
It's an insidious process. What happens is that over years, due to factors like high blood pressure, smoking, or poor diet, the inner lining of those coronary arteries gets damaged. The body tries to heal it, but in the process, deposits of calcium, fat, cholesterol, and fibrous tissues begin to build up within the artery wall itself.
This is the plaque. And as the plaque grows thicker and thicker, it pushes inward, making the lumen the actual hollow tunnel. The blood flows through narrower and narrower. The pipe gets clogged.
And when the pipe narrows, less blood volume can pass through it, which means less oxygen reaches the hardworking cardiac muscle cells downstream. This sets the stage for the third disorder, which is actually a symptom, angina or angina pectoris.
Angina is the physical sensation of that starvation. It manifests as acute, severe chest pain. It happens when the heart muscle simply isn't receiving enough oxygen to perform the work being demanded of it.
Exactly. The muscle shifts to anaerobic metabolism, producing lactic acid, which causes intense pain. Angina can occur in men and women of any age, but it is vastly more common among the middle-aged and elderly whose vessels have accumulated decades of CAD plaque. It characteristically flares up during physical exertion, like climbing a flight of stairs when the heart rate spikes and demands more oxygen, but the narrowed coronary vessels simply physically cannot deliver it. When they rest, the heart rate drops, the oxygen demand falls, and the pain subsides.
But if that plaque totally ruptures and a clot forms, completely sealing off the coronary artery that leads to the death of the muscle tissue, a heart attack. Which brings us to a massive clarification point. People constantly misuse cardiovascular terms. Heart failure, cardiac arrest, heart attack. They are not the same thing.
In medical education, distinguishing these three is vital. Let's start with heart failure. Heart failure is a mechanical pumping issue. It means the state of the heart when it has become too weak or too stiff to pump blood effectively enough to meet the metabolic needs of the body. The engine is failing.
Because the pump is weak, blood starts to back up in the plumbing behind it. If the left ventricle is weak, blood backs up into the lungs, leaking fluid and causing congestion. That's why it's historically called congestive heart failure. The patient slowly drowns in their own fluids.
Yes, heart failure is a chronic progressive decline. Now contrast that with cardiac arrest. Cardiac arrest is an electrical problem. It occurs when the heart's nodal tissue or electrical grid completely malfunctions and the heart abruptly and entirely stops beating. The pump simply turns off. The patient instantly collapses.
And finally, a heart attack, clinically known as a myocardial infarction. This is a plumbing problem. This occurs when a coronary artery gets completely suddenly blocked, usually by a ruptured plaque and a blood clot. The blood supply to a specific portion of the heart muscle is abruptly cut off, and that piece of the muscle rapidly dies from oxygen starvation.
The distinctions are clear. Heart failure means the pump is too weak to keep up. Cardiac arrest means the electrical system crashed and the heart stopped beating. A heart attack means a piece of the heart muscle is actively dying due to a blocked pipe. A heart attack can cause cardiac arrest, but they are fundamentally different events.
That is a brilliant and necessary clarification. Well, that brings us to the end of our anatomical and physiological journey. But before we sign off, I know you have a way of locking this information in.
I do. In 15 years of teaching, I found that you need to distill complex systems down to their absolute core truths to retain them. I call it the mentor's sticky summary. Here is what you must remember. The top 5 indisputable facts from today's deep dive into the circulatory system.
Give it to me. Let's lock it in.
Number 1, blood versus serum, the golden formula. Serum is simply plasma with the clotting factors, primarily fibrinogens, removed. It's the fluid left over after a clot forms. Number 2, the white blood cell hierarchy. When thinking about your immune army, neutrophils are by far the most abundant doing the frontline phagocytosis. Basophils are the absolute least abundant, handling the chemical inflammatory response.
And the mnemonic to remember the descending order of all 5. Never let monkeys eat bananas. Neutrophils, lymphocytes, monocytes, eosinophils, basophils.
Perfect. Number 3, the RH danger. Erythroblastosis fetalis. Remember the timing. The danger occurs when an Rh-negative mother carries her second Rh-positive child because the mother's immune system was sensitized and formed memory antibodies during the delivery of the first child. We prevent it with the RhoGAM shot. Number 4, The valves. You must know the geography. The tricuspid valve sits on the right side of the heart. The bicuspid or mitral valve sits on the left side. Their singular function is to catch the pressure wave and ensure one-way blood flow. And finally, number 5, the ECG keys. Decode the squiggly line. The P wave represents the electrical excitation of the atria. The massive QRS complex represents the electrical excitation of the ventricles. The T wave represents the ventricles resetting. And practically you count the QRS complexes to quickly calculate the heart rate.
That is pure gold. If I had to give a 60-second summary of this entire biological masterpiece to someone who asked what I learned today, it would be this. Your heart is a myogenic, auto-excitable, 4-chambered double pump. It uses a custom internal electrical grid starting at the SAN pacemaker to squeeze out roughly 70 milliliters of blood per beat. That blood travels through a totally closed loop of arteries and veins operating at high and low pressures. The fluid matrix of that blood, the plasma, drops off oxygen, glucose, and nutrients to every single one of your 37 trillion cells while simultaneously picking up their trash. It relies on millions of red blood cells acting as cargo ships, a standing army of white blood cells neutralizing microscopic threats, and specialized platelets ready to initiate a calcium-dependent chemical cascade to patch any leaks in the pipes.
A phenomenal synthesis. It takes a static textbook diagram and brings it to life as the dynamic, relentless machinery that it truly is.
I want to leave you, the listener, with one final, slightly provocative thought to mull over today. We spent this entire time marveling at the heart. We established that the cardiac muscle is the ultimate engine responsible for pumping life-giving, oxygen-rich blood under massive pressure to the entire macroscopic city of your body. It holds the ocean of life inside its massive chambers, but it cannot absorb a single drop of that oxygen from its own internal chambers. The walls are too thick. It requires its own dedicated, tiny external plumbing system, the microscopic coronary arteries wrapping its surface just to survive. What does that say about the extreme vulnerability of this seemingly invincible system? The massive engine that feeds the entire body is entirely fundamentally dependent on its own smallest, most delicate supply lines remaining perfectly unblocked. If one tiny pipe, just a few millimeters wide, clogs with plaque, the whole city falls. It is incredibly robust, yet terrifyingly fragile.
It is a profoundly humbling thought. It highlights exactly why understanding this physiology and making choices to protect that delicate vascular lining is perhaps the most important thing we can do for our own longevity.
Absolutely. Thank you for joining us on this exploration of the microscopic metropolis inside you. Keep questioning, keep visualizing the mechanics beneath the skin, and keep digging into the incredible, relentless machinery of your own body. Until next time.